let's move on to the next speaker, Dr. Vinod Scaria. Dr. Scaria is a pioneer in precision medicine and clinical genomics in India. He is best known for sequencing the first Indian genome. He now works at the CSIR Institute of Genomics and Integrative Biology, as well as an adjunct faculty at the Indraprastha Institute of Information Technology, Delhi. Among many activities, he also contributes to the COVID-19 study, which includes the establishment of the COVID-19 open research data and resources. Today, the title of his talk is Genomes from Personal to Populations and Back. Dr. Vinod Scaria, please.
Thank you, Professor Nakai, for the great introduction, and good morning to everybody. It's a pleasure to be here. Over the next 30 minutes or so, let me get into the talk. Before that, let me introduce and make some disclosures, because many of the work that we do have gone into licensing agreements with a number of companies. So before I start my talk, let me also give you a brief background of what we do. We call ourselves people who work on genome informatics. That necessarily means that we work on computers trying to analyze genomes, and we try to ask a number of pertinent questions trying to do this. We try to address the non-coding RNAs in terms of their function and in terms of the epigenetic architecture. We try to work with a large number of clinicians as part of multiple different programs into Mendelian genetic diseases, as well as pharmacogenomics. And that's what I'm going to talk to you today. We also work in the area of population genomics, working closely with a large number of country-wide programs in Southeast Asia and the Middle East. We were also the founding members of the Pan-Asian Population Genomics Initiative. And a small part of my lab works in the area of pathogen genomics, mostly in the area of tuberculosis, and very recently in looking at viral epidemics. Now, to give a brief background on why we were very fortunate to be in the right time, in the right place, to be able to do this is exemplified in this particular chart. As you see here, the decade that followed the human genome draft sequence, or the first human genome, as rightly called, has seen a drastic reduction in the cost of genome sequencing. And we were in the right time. I started my lab in 2007. And as you see here is also the time when the cost of genome sequencing was dwindling very rapidly. And the outcome of this was quite remarkable, that we could now afford to do sequences not just of one human genome or the reference genome, but probably numbers of human genomes in small labs like ours. And that's contributed a large number of public genomes that are available today. And to pose this question, way back in 2008, we looked at the human genome diversity. The map was provided by the Human Genome Diversity Program in terms of the human admixture across the different continents. And if you plotted the genomes, so the whole genomes that are available at that point of time, the 6 at that point in time, this included the reference genome, the Craig Venter and the Watson genomes, and the Chinese, Japanese, and the Korean genomes, which came from all quite homogeneous or less admixed populations. And what you see here in the middle of the slide is a large gap, which probably encompasses a third of the population of Asia, which includes quite admixed populations in the Middle East and the Central and Southeast Asia, which didn't have quite any genomic representation. So that's essentially when we started. In 2008-2009, we put together the first genome in India. And the idea of doing this was essentially learn the tricks of the trade on not just how to sequence the genome, but also to be able to put together the analysis in terms of understanding what are the functional variants in the human genome. Now, it was just one, but we quickly went ahead and collaborated with a number of countries in and around this region to also do the first Sri Lankan genome in 2011. We were also participants in analysis of the first Malaysian genome in 2013. And we were one of the founding members of the Pan-Asian Population Genomics Initiative which aims to look at the diversity of population genomes in this part of the world. In the lab, we also hold two large databases, SAGE for the South Asian genomes, which today encompasses 1,200 exomes and genomes, and Almina for the Middle Eastern populations, which today encompass 2,000 whole genomes and exomes from the region. And quite very recently, we embarked on a population genomic initiative, and at least in a pilot scale in India, to complete 1,000 whole genomes from India, which is part of the IndiGen program. I'll talk to you about this later in my slides. So the question is not anymore whether you can do human genomes, but how do we really put that into application or put that into clinical practice? And that comes to the problem of rare genetic diseases. And in fact, many of these conditions have been known for ages, but of course with the wrong connotation as monsters or sinners. And as you see here, this is actually the pictorial representation of the monster of Ravenna, drew way back in 1500s. But today what we know is that many of these are genetic mutations which give abnormal physical presentations of individuals, and otherwise they are rare and undiagnosed conditions. Now to put this number in perspective, there are around 300 million people in the world who suffer from one or the other rare genetic condition or a rare genetic trait. And India, we have close to around 70 million people with one or the other of these extremely rare conditions. There are around 3,500 odd rare conditions reported in literature, and at least 1,500 of them do not have a gene or a genetic locus yet annotated. And that's essentially a big problem. Apart from that, many of these patients go through multiple physician visits, approximately 8 to 9 physician visits, 3 rounds of misdiagnosis, and take approximately 7 years before even they have a provisional clinical diagnosis. And that's largely because of the lack of awareness about genetic diseases, the lack of availability of affordable system for genetic screening. And even if you were to do screening, there's a lack of understanding about the mutational spectrum of pathogenic genetic variants in a population. Now, of course, that could also be an opportunity because we are a large population. India encompasses a sixth of the world population today, and our genetic population is quite diverse and stratified into endogamous groups. And that's pretty much true, not just in India, but whole of Southeast Asia. And more importantly, we have close-knit family structure. And unlike the rest of the world, which was displaced in the last 1 century by multiple wars, famines, and infectious diseases, India has largely been untouched by many of these world events. And we still have a close family structure. And in many ways, most of the individuals could trace 3 to 4 generations of their family back to their ancestral village. And that essentially is a central point in genetics, because all of genetics starts with an extended pedigree. Now, 5 years ago, to be exact, in 2015, we put together this program called the Guardian. The Guardian stands for Genomics for Understanding Rare Diseases, India and its network. And the idea was very simple. We would work with clinicians who would see undiagnosed or rare conditions in the clinic. They could follow a standardized process for consenting sample collection, and we would do a standardized genome or exome sequencing, data analysis and reporting. And whenever we find a mutation, we return it back to the clinician as a genetic report. But that happens only in approximately 50% of the times. The rest of the times, we need to do much more investigations, including in vitro, in vivo, and in silico validations to be able to prove that the genetic variation caused the disease. And that's essentially the basic science part of the entire program. But eventually, the idea is that we will be able to prove that a particular genetic variation caused the disease and return it back to the clinician so that he can make clinical decisions and act upon it. And today, this program encompasses 280 odd clinicians from 70 major clinical and medical centers across the country, making it one of the largest rare disease programs in this part of the world. And it's not just diagnosis and research, but also to implement policies through education and, of course, working with a large number of colleagues And this is one of the recent papers that we have sort of talked about. And behind all this program is a large informatics system built over almost a decade, which not just includes the computational part of the infrastructure, but also data sets which were curated and collected over almost a decade. This includes supercontrolled centenarians who are individuals over 100 years of age, but without apparent disease. This includes normals and family controls, and very recently the population scale data from the IndiGen program. And I will give a few examples of how this program works with a few clinical cases to exemplify a few points. Now, this is one of the first families who came to us and referred from the D.Y. Patil Hospital in Pune. And as you see here, the two children with abnormal skin born to apparently normal parents. As you see here, the parents have normal skin. And when they came to us, they also came with a karyotyping report, which suggested a chromosomal abnormality. But of course, as you see here, the abnormality was also inherited from one of the parents, while the parent was apparently normal, which sort of excludes the causative nature of this particular chromosomal abnormality. Now, this points to two things which are very important. One, it is a disease, lamella-ichthyosis or congenital lamella-ichthyosis, an extremely rare genetic condition. The second, there are two children born of the disease. That necessarily means that the genetic counseling services were not really appropriate. To have another child born with the same recessive condition as in many ways showing the lack of genetic counseling. Third, there was an inappropriate genetic test administered, which is karyotyping, because congenital lamella-ichthyosis is not caused by chromosomal abnormality, but nevertheless caused by simple nucleotide genetic variants in around 9 genes. So this is inappropriate genetic testing when it is available. So what typically we do to such families is to do exome sequencing. And we did exome sequencing and we did find a mutation in the child. And you could go back and validate this mutation on Sanger capillary sequencing. And what we show here is that for the children were homozygous for the genetic mutation. And like all families who come to us, they underwent genetic counseling and they had a prenatal testing done for the next child. And the next child was heterozygous and a normal child was born way back in 2015. Now, to add happiness to this family, people with TGM1 mutations with lamella-ichthyosis respond very well to retinoic acid. So both these children were put on retinoic acid, and that's a photograph of these children after 3 months on retinoic acid. And as you see here, the hair has grown, the face is cleared up, and if they were fully clothed, nobody would ever recognize that they have a genetic condition. This is another family and a diagnostic DC. The child was referred to us from the All India Institute of Medical Sciences with a query mitochondrial genetic disorder. We did a mitochondrial sequencing, but we didn't find a mutation. So we quickly went and did an exome sequencing, and for which also we could not find a genetic mutation. But unfortunately, the child had died by that time. But nevertheless, the samples were preserved so that we could do a whole genome sequencing. And the whole genome sequencing did find a genetic deletion, approximately of 142 kilobases, which is homozygous in the child. And in the next pregnancy, of course, they did want to do prenatals, and we did recognize a homozygous deletion in the product of conception, sort of suggesting that the child would also be affected by this disease. But before that, we needed to really prove that this genetic deletion caused the particular phenotype of mitochondrial disease, because this was a chromosomal abnormality and not a mitochondrial genetic disease. So what you could quickly do is to either take fibroblasts from the skin of the child, culture them, and do mitochondrial assays, and also build model systems where the particular gene or deletion could be made in vitro using genome editing tools. So in 9 months, what we could do is to prove that the particular genetic deletion caused the particular phenotype. And apart from that, also build a genetic diagnostic, which is more sensitive and more cheaper than whole genome sequencing, which is based on MLK, which is very effective in making diagnosis about chromosomal deletions. This is the third family, and this is from a place called Agra. And this family petitioned the President of India asking to euthanize all of the 6 children who were affected from a rare genetic condition and a progressive neurological disorder. And the children in the clockwise direction are from the youngest to the oldest. And as you see here, the oldest kids are practically bedridden and in a vegetative state. And this progressive neurological disorder starts around 3 years. As you see here, the youngest child is still unaffected. The child can still stand up and talk to you. But nevertheless, over years, it progresses into complete debilitation without any diagnosis. So like all other families, we do an exome sequencing of multiple members of this family. We did find this mutation as MLC1, confirming the diagnosis of cystic megalencephaly, or the longer name is megalencephalic leukoencephalopathy with subcortical cyst type 1, an extremely long name and extremely rare genetic condition affecting 1 in a few million individuals. Now, what we did is to actually go back and look at the family, the extended family. We could draw a pedigree, and just by drawing a pedigree, we could find another affected member in the family who was also tested positive. And since the pedigree, as you see here, suggested consanguinity, which is also very common in Southeast Asia, we went back and did a family survey, a community survey rather, and we found another 2 families with the same genetic condition and the same phenotype. So this was very surprising because this was otherwise an extremely rare genetic mutation, and this mutation was previously reported from 1 patient from Persia. And this family survey, the community survey, revealed us something very stark. Approximately around 28% of the individuals in this close consanguineous family of community was actually carrier for this extremely rare genetic condition. And we found 4 families in total in this community with this genetic mutation. Now, the power of genomics is that right from the case referral to the diagnosis, to the publication, to the diagnostic and market, you can actually cover this entire cycle of diagnostic odyssey in approximately 2 to 3 years. And today this diagnostic is available, also named after the community, because a lot of people from this community could then avail this genetic diagnosis, not just for the diagnosis, but also screening for potential partners if they were to marry within the same community. And incidentally, this individual many years or many generations ago, to be exact 52 generations ago, had migrated from the Middle East into India. And that was also the right time when we were working with a lot of Middle Eastern counterparts to create this database, which is one of the largest of genomic databases of the Middle Eastern and North African populations. And it is not just one single family who migrated from the Middle East into India, but today there are many, many more communities which trace their ancestry from the Middle East into India. While there have been Indians for many generations, in fact, many centuries, they carry in their genome a small part of their genome from a different population, and then therefore would be predisposed to different sets of diseases, genetic and otherwise. And then therefore, it is very important not just to understand the genetic origin of the genealogy of these populations, but also to be able to educate the individuals who take care of them, who are essentially the clinicians, so that they can be very well informed in advance about genetic diseases, which are otherwise prevalent in a different part of the world, but now prevalent in a small community down here in India. And that's where we worked with a lot of collaborators in the Middle East to create DALIA, to create one of the most comprehensive resources of genetic diseases in the populations. But what's really more important is to bring to you this fact that the conventional practice of genetics has always relied on the fact that the patient will seek help and come to a clinician for a genetic diagnosis. But that concept by itself is not always true, especially when the disease is not debilitating, has only some functional abnormality, and of course is associated with a taboo. And this is one such family which is extensively investigated by our collaborator, Dr. Sandeep Mathur. And this is hypogonadotropic hypogonadism. Except from the fact that the individual is sterile and have sexual dysfunction, the individual has no other functional abnormality and therefore very unlikely to seek medical help. And then therefore, practically all estimates of genetic diseases in a population or a community could be actually wrong if you rely on this fact that patients with genetic diseases will come to a clinician. So what's the option to understand genetic diseases in the community? The only way is probably to go back and sequence a lot of normal individuals or apparently normal individuals in the population. And that's how we initiated the IndiGen program to sequence 1,000 individuals representing major populations across the country. And the idea is to create a comprehensive allele frequency resource, understand prevalence and founder effects, and also a pharmacodynamic landscape of the population. And of course, create systems for analyzing genomes at clinical turnarounds and clinical scales. Now, I'll give a few examples of what this data has brought about. And one of these examples that I'm going to talk about is of treatable intellectual disabilities. Now, intellectual disability in children is something very prevalent, but what is really not known is that a significant large subset of intellectual disabilities are treatable. And today we know about 81 genetic disorders caused by mutations in around 104 genes, which are treatable, or in other words, there is a specific management protocol or a treatment protocol available for these conditions. So in other words, early diagnosis of these conditions would allow many of these children to be treated and therefore avoid intellectual disability later in their lives. So what we did was very simple. We could go back to a database like HOMIM, pull out all the diseases and the genes. You could go back to literature and curate a large number of genetic variations which are reported for these genetic disorders. Go back to the population scale data and ask this question: what are the variants which are prevalent in the population? In other words, therefore, you could know what is the prevalence of known genetic variation. Of course, the caveat is that you would miss genetic variations which are novel in the population, but nevertheless, that is understandable. And now ask this question: what are the variations which are prevalent in the population compared to the rest of the world? And what we found was quite remarkable. We found a large number of genetic diseases which we didn't expect in the first place. For example, Wilson's disease, the heterozygosity was 1 in 147, phenylketonuria and cerebral xanthomatosis, the frequency was 1 in 171, citrullinemia type 1, the frequency was 1 in 206. Now just think of it in this way. If you now start newborn screening for many of these disorders for which newborn screening doesn't exist in the population, you will be able to diagnose them much earlier and be able to treat them. And if you think this is just numbers which is in the population, it is not really true. Even in a cohort of undiagnosed genetic conditions, just by using this detail of annotated genetic variations, we could figure out patients which are previously undiagnosed for these conditions. And today we have a genetic screen which you can now apply for a population in a quite affordable and accessible format, which now covers around 1,700 diseases, 1,300 genes, and 10,000 genetic variations, including the genetic variations which are very prevalent in the population. Now, there are many other diseases which don't really require clinical attention because they are not really diseases per se, but they are genetic traits. One of the examples is rare blood groups. They're very, very prevalent in the population. Approximately 1 to 2% of individuals in any population would have a rare blood group. Now, that should not really affect you because unless you are in a position to receive transfusion, you would not really care about a rare genetic disease or a trait rather, like a rare blood group. Now, the idea is you could look at the population scale data to understand where are the rare alleles in the population and be able to build genetic diagnosis which are very cost effective and accessible for the population so that we can plan and create rare blood group registries so people who have rare blood groups can now access a match for another individual who is ready to be a donor for such an eventuality. So this is what is possible in case of looking at population genomes. And it's also important to communicate the genetic information back to the patient. And every individual who are participant of the IndiGen program for public health genomics today has a genetic card which can now communicate information in a relevant format for a few things like pharmacogenomics, like incidental findings in terms of the carrier status, and of course, some fun facts which are not of clinical relevance, but of course, things like that you can drink a lot of coffee or you can smell a particular substance and so on and so forth. So in other words, you also need to create systems to enable secure, privacy protected, scalable, and of course, point of care genetic reporting, which is clinician enabled, because there's a lot of genetic counseling that needs to be embedded into the entire architecture. So in summary, you could start from looking at genetic diseases, and that's what I talked about Guardian. We also have a genetic service, which is run by my colleague, Dr. Mohammed Farooq, which today gives 130 genetic tests, which is called as GOMED. We also actively engage in clinician training through the Sanofi fellowship programs for clinical genomics. And by doing this over, you also identify not just genetic diseases, but also build diagnostics, which can now go back into the diagnostic industry. We have a large number of such diagnostics in the market with multiple different partners. Now, by doing all this, you also increase the reach of diagnostics. That means you're diagnosing more patients. And for all of these patients, you would need to also delve into discovering new therapeutics. And that's where we use a lot of our analytics skills, a lot of colleagues and our expertise to build new diagnostics and new therapeutics. And of course, by population scale genomics, you can accelerate all of these by providing the scale and reach of many of these genetic diseases which are previously unknown in the population. So in summary, genomics can make medicine very predictive. You can do genetic screening, pre and postnatal screening. You can make medicine very preventive by preventing genetic diseases from occurring in families. It can make medicine very precise by offering early and accurate diagnostics. And it can make medicine very personalized by offering them the right therapy. But this is all possible only with the last P, and that's what we call as the central P, which is essentially the participation, not just of patients, not just of clinicians, but also a whole society of people around patients and families. And I think I'll stop there and take questions, but before that, let me acknowledge a lot of people who made this possible, a lot of students and faculty who collaborated quite extensively. Dr. Sridhar, who has been a quite good collaborator for almost a decade and has been a partner in crime for almost all that we do. Fantastic collaborators from across the country, from the Guardian Consortium, and of course, large cohorts like Dr. Sandeep Mathur and others. International collaborators, Dr. Wajira and Mohammed Zaki Saleh from the UITM in Malaysia. And of course, this is all possible with consistent funding for the last 1 decade from CSIR. I think I'll stop there and take questions, and I'll spend probably half a minute trying to advertise the new thing that we do. In light of the pandemic, they have been involved in a lot of open data and open resource initiatives, mostly in the area of bioinformatic analysis, in genomics and epidemiology data. And if there are anybody interested, feel free to contribute. Thank you.
Thank you. Many thanks, Dr. Scaria, for excellent talk. So we still have about 10 or more than 10 minutes for questions and answers session. So the audience almost already some questions appear in the chat. Could you, Doctor, could you check the chat session?
Yeah. Can you share what kind of training consultation that we could do for clinicians in order for them to learn about how to utilize genome information when consulting patients? Yes, indeed. I'm sorry I didn't put a slide for that. So we do quite extensive training programs for clinicians. This includes short-term CME programs to in-depth programs, like for example, we have been running the clinical variant interpretation and analysis course, which is a 12 weeks online course for clinicians so that they can interpret a genetic variant according to the ACMG/AMP guidelines to call it as a pathogenic or a benign, so on and so forth. So if you are interested, you could just mail me. I would be more than happy to provide you more information. And the second question is, how about calculating the polygenic risk score for genetic diseases among Asian population? Until now, only 10% of the genomic data are studied of Asian origin. Yeah, so indeed, this is quite important. Now, polygenic risk scores are typically for complex disorders, which is not typically what we work on. We work on Mendelian genetic disorders. In many ways, you're working at one genetic variation, which has a very large effect size and can cause the disease. But having said that, of course, polygenic risk scores, even in terms of genetic diseases like Mendelian genetic disorders, are quite relevant, because even very common disorders like sickle cell anemia, which is worldwide caused by just one single mutation, has an enormous phenotypic diversity, and this could be contributed by many other modifier locus, and then therefore polygenic risk scores for genetic diseases are important. Yes, there are only 10% or probably much lesser of the data in global scenario contributed from Asia. And obviously, over time, we foresee a lot of countries would take up population genomics initiatives. And of course, this is probably going to change the numbers, at least in the next 1 decade, if a lot more countries take part in population initiatives. Okay, so is there any evidence for LNC RNA to be a reason for inherited genetic diseases? Yes, there are a few. If I'm not wrong, less than a dozen of diseases which today are known to be caused by either deletions or genetic mutations of long non-coding RNA. So non-coding RNAs are indeed important as a causal factor for genetic diseases. But more importantly, they could also be a significant modifier locus for many of the Mendelian genetic conditions. There's another question. Could you also share what kind of challenges that you face when collecting samples and data? What about data protection issues? Typically, we do not have a lot of issues with sample collection and data, because all these studies are approved by our institutional ethical committees, as well as the participating institutes' ethical committees. And in many ways, this also includes the permissions to actually recontact the participants, because these are conditions which are genetic in origin. In many ways, they are undiagnosed. So it is in many ways a lot of interaction with the clinician back and forth on the phenotypes and the genetic analysis. And in many cases, you will also need to do additional investigations for these patients. So since this directly benefits patients in the first place, this has been quite, and patients have been quite forthcoming to contacting me back, but not really sure about the population genomics initiative. There are many patients who have decided not to be contacted back when they have provided us samples. Okay, so there's a question. Consanguinity is a huge issue. I'm sorry. Consanguinity is a huge issue in several populations. Would India be willing to mandate genetic counseling prior to marriage? Now, mandating anything is essentially a social issue because there are a lot of ramifications to it. But at least the most important thing that needs to be put in place is, first, there need to be awareness about genetic diseases, not just among the patients, but also among the clinicians. In terms of genetic counseling, India doesn't still have the resources to mandate genetic counseling for that matter, because there has been only one accredited master's level program in genetic counseling in the entire country. And many diploma courses which are non-accredited as far as the rule goes. So eventually, the starting point would be the patients and families and their close community, which is onus on the clinician and the genetic counselor to work upon and counsel. But as awareness and genetic testing becomes widely available and accessible, yes, eventually that would be the best and the cost-effective way to prevent many genetic conditions. There's a question on current pandemic has drastically changed the pathogen molecular diagnostic capability in remote parts of India. How can India make use of the development to drive improvements in screening and diagnosis of genetic diseases? Yes, in many ways, the pandemic has really changed the accessibility of genetic tests, at least in the last 6 to 8 months in many parts, especially in the rural and remote areas. Now, there's nothing that can be immediately changed, but we are all hopeful that the pandemic will eventually wane out, and then people will be able to access many of these genetic tests. There have been a lot of initiatives on building point-of-care genetic diagnosis for prevalent genetic diseases, the likes of sickle cell anemia, thalassemia, which can now be implemented in rural areas. And there have been a large governmental program also mandating the development and application of such tests for the prevalent genetic disorders. I think that are the questions. If there are many more questions, you could put it in the chat box, or you could mail it to me. I'd be more than happy.
Yeah, thanks. It is about the time. But before concluding the session, could you give some comments about the situation of the COVID-19 in India?
Okay, so looking at the general new cases over time, the cases have been dropping over the last 2 months. Now, taking about very specific cases of cities or metropolitan cities like Delhi, we are going through probably the 3rd wave in terms of the numbers. And at this point in time, we are clocking almost 7,000 to 8,000 cases every day in places like Delhi and Mumbai. So in metros, the cases have been coming up in waves, but in totality across the population, the cases have been dropping over the last 1.5 months.
Thanks. So thank you very much. I'd like to close the session, but also there may be other questions that I think maybe Doctor has answered most of the questions, and maybe a bit.
Professor Kenta, there's one more question at the chat.
Okay. Dr. Scaria, could you ask another question? That there is an
yeah. So
sorry.
Svante Pavo recently said that there's a gene cluster on chromosome 3 is a risk factor for severe COVID-19, and that gene cluster has a Neanderthal origin, and 50% of South Asians have this gene cluster. My thoughts. Okay. Now these are so if you look at the severity or the genetics of severity of COVID, it encompasses a wide spectrum, right from monogenic conditions in the interferon pathway. If you were to look at these genes, there have been a series of papers from Rockefeller University on monogenic causes which cause severe COVID, especially in the young individuals. Then there's a spectrum of genetic events into the complex space, and many of these risk factors that are sort of risk factors by assumption, they're not really causative, but they're essentially risk, are essentially small risk, typically of odds ratio of 1.1 or actually less. Now, in a population scale, this would not really make a huge difference because there are many other factors which we do not account when comparing populations. Like, for example, one of the things that we need to compare is, for example, cross-reactivity to other pathogens. Now, that is very different between the East and the West, or the East and the South. Now, genetic risk factors per se of this order of magnitude, say typically odds ratio of 0.1 or less, in a population scale is not going to really contribute to more severe disease as suggested in most of Southeast Asia. And if you were to go by the numbers, actually Southeast Asia is rather much protected from severe disease or mortality if you adjust for the population size compared to many of the countries in the West. Now, the reasons for this is really do not know. But having said that, there are many more risk factors which are yet to be uncovered, especially from association studies from Asia, which I have not yet seen any in literature.
So thanks again for a great talk. Let's close the session. We can't upload. Sorry, but let's close the session. Thank you very much.
Thank you very much.